Good morning, my name is Jane Bayu, your regular biologist. You're welcome to my channel. We'll be looking at the 2026 biological particle questions. We'll be looking at possible questions that will be asked and we'll be analyzing the specimen. We'll be starting with granite pod. So the first question I wrote here is identify specimen A. In the exam, you make sure that you get your pen, you get your pencil, and you write some of these names in capital letter if you know you have a small handwriting. So let's start with specimen A. Identify specimen A, granite, or type of fruit of specimen A. It's a legume. Reason: it contains 2 to 3 seed chambers. 2, it is formed from one carpel. And 3, the seeds are arranged along a margin. Name one pest of specimen A. A pest of specimen A. The answer, I have 3 here. Aphids, caterpillar. The caterpillar feeds on the leaves of the grass. Or one disease that occurs in specimen A is called granite rosette. Then the causative agent, virus. Classification of specimen A. It's a true, simple, dry, indehiscent fruit. If you're now asked what's the reason for classifying it that way, the fruit pericarp do not split to release the seeds. Type of germination of specimen A and the life cycle. It's an epigynous germination and an annual crop. Type of placentation found in specimen A. It's marginal. If you are asked reason, the reason is that the ovules are arranged in a single roll at the margin of the ovule. Mode of dispersal of specimen A. When you're writing animals, humans are correct, but you don't put all, you don't put the 2, you put 1. So if I say humans, with reason, humans, dog, harvest, eat, and plant the seed. For animals, animals also dog, eat, and discard the seed. Biological function of specimen A. From the plant it was obtained. The thick pod prevents water from going into the seed, for seed dispersal, for seed storage. It fixes nitrogen in the soil. Any of these are function, and is also a source of nutrients, example vitamin. What's the relationship between specimen A and B? Specimen A is a fruit that encloses specimen B, or you can write it in this form. Specimen A is the fruit that is developed from the ovary enclosing specimen B, which is formed from the ovule. You know, everything just demands your English. So we go to specimen B. On specimen B, we'll be drawing a table. We have procedure, observation, and inference. I want us to know that if your procedure is wrong, it affects your observation and inference. So, and when you are writing the procedure, you make sure that everything you did in the lab, you analyze them. Example, crush specimen B was smeared on a paper. You write it in English in full, not specimen B smeared. No, you write it in full. A small portion of specimen B on a white paper and observe. Then under the observation, translucent mark seen. Under inference, fats and oil present. And the second question I wrote here, add a few drops of iodine solution to specimen B on a white tie. Observation, blue-black coloration observed. Inference, starch is present. Name 2 elements found in the food class of specimen B. The answer is carbon, hydrogen, oxygen. Economic importance of specimen B. Number 1, it fixes nitrogen in the soil. It helps to add nutrients in the soil through crop rotation. It forms livestock feed. Example, groundnut cake and groundnut hill. It's a good source of food. It's also a source of foreign exchange, and it's also a source of employment. Any of them is correct. Function of food class in specimen B. What's the function? It helps in the formation of cell membrane. It is oxidized to form energy. Stores under the skin to help reduce heat loss, and it's a good form of food reserve. Specimen C, D, and E. Specimen C is a beaker with 20 ml of water. Specimen D is beaker with 20 ml of table salt solution. Then specimen E is beaker filled with sandy soil. Procedure, observation, and inference. So under the procedure, measure, let's say variably, measure 20 cm of sand in a beaker. Then add specimen C, the 20 ml of water, into the sand in the beaker. Now I wrote down a question. What do you observe as you add the water? The answer is air bubbles was observed. And the second question I wrote here, of what use is your observation to the soil above? The soil contains large air spaces. So when that air bubbles occurred, it aerates the soil, number one. Number two, it makes the soil porous. Suggest a suitable title for the experiment. Experiment to show soil porosity. Experiment to show aeration of soil. And number three, experiment to show soil contains air. Specimen F. Portion of alimentary canal of a small mammal from oesophagus to the end of a stomach, neatly placed on a dissecting board. First question here might be to draw. Observe and draw what you can see. And in drawing, make sure that you put your title. You must show the oesophagus. You must show the diaphragm, because I believe that the oesophagus is a long tube that runs into the diaphragm, connects to the stomach, and then stops there, because that's the end of the stomach. So the question, and then when you're drawing, you must also label, use straight lines in ruling, and then you make sure that your labeling points to what you're talking about. Mode of nutrition of specimen F. It's herbivorous. Mention the kingdom, class, and phylum name of specimen F. Kingdom is Animalia, class is Mammalian, and phylum is Chordata. On the dissecting board, observe specimen F and state 5 organs seen. I wrote down esophagus, the diaphragm, the cardiac sphincter, the stomach, in order of arrangements. Let me go again. The esophagus, the diaphragm, the cardiac sphincter, the stomach, and the pyloric spleen. So digestion of the cellulose starts in the small intestine, when the bacteria in that cation helps to digest cellulose. So in the stomach, the only digestion that takes place there is protein digestion and temporary storage of cellulose. So there is no digestion of the cellulose. Type of food consumed by specimen F. We consume dry grass, cellulose, and hill. State 5 observable structural features that adapt specimen F to their habitat, or that adapt specimen F to their mode of feeding. Let's start with the esophagus. The esophagus is a thick, muscular, long tube that helps to transport food and water. The diaphragm is a thin, muscular sheath that separates the thoracic segment from the abdominal segment for during breathing. The cardiac sphincter, it's a thick ring of tissue that prevents food from going up. The stomach. The stomach is a large muscular J-shaped sac that temporarily stores cellulose, and it also helps in protein digestion. The pyloric sphincter. This is a thick muscular valve that regulates the food as it enters or it passes to the small intestine. Specimen G. Identify specimen G. African mistotto. Okay, we asked to make a drawing of specimen G. What I feel that it should be important there is that the host must be shown. The attachment where the mistotto uses its hostorium to attach to the host must be shown. The fruits must be shown, and the flower and the leaves must be shown. So if you draw it, these things can carry good mark. Botanical name of specimen G is called Tapenanthus lauranthus. State structural features that adapt specimen G to its habitat. Number 1, swollen root-like hostorium for penetrating the host bark to absorb water and minerals. Leathery leaves containing chlorophyll for photosynthesis. 3, leaves are thick and wasky to prevent transpiration. Sticky fruits for seed dispersal. Bright tubular flowers, in bracket red in color, for pollination. Swollen nodes for producing more hostorium that enables it to attach to the host plant. Question: one significant feature that distinguishes specimen G from other parasites: the ability to possess green leaves containing chlorophyll to make food. Mode of dispersal of specimen G is the beds. Reason: the fruits are sticky, containing seeds. So when the birds feed on the fruit, the seeds are sticky with sticky gel. They stick to their beak, the beak of the bird. So when the bird flies to or has flown to another plant, when they get to the stem, they scratch their beak on the branches. So with the help of the sticky gel, the seeds stick to the new stem of another tree, and then they develop hostorium that continues the growth of mistotto. To specimen H, we're going to draw a table as normal, procedure, observation, and inference. Evaporated milk poured in a beaker and labeled milk. Pour 5 cm of specimen H plus 3 drops of sodium hydroxide plus 3 drops of copper 2 sulfate bit by bit. Now, when you add the sodium hydroxide, you shake. Then few drops of copper 2 sulfate, you put it drop by drop and you shake. Observation: purple coloration observed. Inference: protein expressed. Name of test performed: Mellon's test or Buret's test. Class of food of specimen H: protein. Function of the named class of food in specimen H. What's the function of protein? Helps to repair worn out tissue. It helps to build up the body, formation of antibodies, formation of hormones, helps in growth and development. So we go to specimen J, fresh fish. So we'll be drawing the lateral view of specimen J. In your diagram, the mouth should be a doubled layer. It's in the picture. You must show the scales, the fins must be shown, the pectoral, the paired fins must be shown, and the unpaired fins. And then the streamlined body is the fish streamlined, must be shown. Class of specimen J, Pisces. State reason for placing specimen J in that class. Presence of scales, presence of fins, presence of a streamlined body, presence of an operculum, or gill cover. Remember, you don't put or, just choose one. State the structural adaptation of specimen J for escaping predators or for survival. The answer is counter shading. That is, the dorsal part is dark gray, while the ventral part is silver white, helping for camouflaging and protecting the fish. External features that adapt specimen J to its environment. So streamlined body for easy movement, caudal fins for easy steering, the dorsal fins, that's the unpaired fins, example dorsal, ventral for stabilizing the fish. The operculum helps to cover the gills for respiration or breathing. The lateral lines for detecting vibration. The scales overlapping. Remember, did I mention when you're drawing, your scales must overlap, meaning that they will just fold by each other. Check your textbook. Overlapping scales for protection and least resistance during swimming, and then the mouth for taking in water containing oxygen. Class of food provided by specimen J is protein, fats and oil, and to specimen K, the maize grain. We might be asked to draw, in 3 of them, the alimentary canal, the fish, and the maize grain. One diagram might come up. So, in which part of specimen K is food stored? The food is stored in the endosperm. The food is also stored in 2 parts, in the endosperm and the cotyledon. They say name 2 parts, but the major part where food is stored is the endosperm. The cotyledon is so small, it does not store food. It houses the radical, there you see the embryo. Phylum and class name of specimen K, Agnoleuophyta or Trachyophyta or Angiospermatophyta. Choose one. Class of specimen K, Leopsida or Monocotyledone. You choose one. How can specimen K be stored? It's a dry maize grain stored in silos. Type of germination and conditions necessary for germination of specimen K. The type is hypogeo germination. Conditions include water, moisture, suitable temperature, and viable seed. Agricultural classification of specimen K. Cereals. Specimen L, M, N. So I'll take the 3 together. L is tomato paste in a petri dish. M, fresh tomato fruit. And N is rotten tomato fruit. Have you subscribed to my channel? If you have not and you keep enjoying what you see, support your girl. Hit that like button, hit the subscribe button, so that when I post any video, you'll be first notified. Thank you. I know you just did that. The type of fruit of specimen M. So I just went with a fresh tomato paste, tomato fruit. Type of fruit of specimen M with reason. It's a berry. Reason, it possesses thin seeds and the endocarp and mesocarp are fused. Which biological measure gives rise to specimen L? I wrote down here drying. So we have procedure, observation, and inference. Dissolve specimen. Dissolve your tomato paste, specimen L, in 5 ml of water. Then you sieve. You sieve it through a filter paper to remove the particles. You take the filtrate, pour it back into a test tube. Then we now test for a reducing sugar. Tomato contains glucose and fructose. So what do? Using our Benedict solution, we add 2 ml or 3 ml depending on what we are at. But the more you add, I think 5 ml should be okay since we used 5 ml of water. 5 ml of the Benedict solution, and then you put it in a water bath, because Benedict solution will not go directly in the heat. It contains alcohol, so you put it in a water bath. The color changes from blue to yellow to green and finally to brick red. Possible test for glucose showing that reducing glucose, which is in a reducing sugar, is present. Placentation of specimen M. It's axial placentation. Studies specimen M and N. That's N is a rotten tomato fruit. Study them without reason. Name the organism responsible for the damage observed in specimen N. The answer is bacteria. You can choose any one: bacteria, mold, fungi. Any of them is correct. Name the disease of specimen N. It's called bacterial weight of tomato. List ways by which each of specimen M and N can be properly stored. Drying. It helps to dehydrate and then preserve the food. Canning. It helps to increase the shelf life. And then freezing. It helps to lower and slow down the growth rate of microorganisms. Take 2 other examples of the type of fruit to which specimen M belongs: garden egg, guava, lemon, lime. I put here orange, citrus. State the food classes present in specimen M. What are the food classes? They include vitamins, minerals, carbohydrates, and water. Observe the difference between specimen M as the firm tomato and specimen N, the rotten tomato. Specimen M, firm to touch. Specimen N, collapses or mushy or soft to touch. Specimen M, bright red in color. Specimen N, dull red in color. Specimen M, smooth skin, no cuts. Specimen N, wrinkled skin with holes and cuts. Specimen M, no water visible. Specimen N, water is seeping out or water is present. So I believe it will not get to this number, but I can give you more. Specimen M, mild, pleasant tomato smell. I can imagine that. Then rotten tomato, foul smell due to decay. Then specimen M, no white particles. Specimen N, white particles seeping out or seen. Specimen M, juicy inside when cut. Specimen N, watery, leaking juice, slimy. One thing there is that if you're drawing the tomato, if you draw the circle, you must make it double, showing the soft epicap. It must be double, remember. Then the placenta must be labeled. You must label the seeds. Seeds should not be just one. We should have like 3, 4, 5 seeds, depending on your diagram, but we cannot have just one seed. Then make sure that where you label point what you are saying. Sometimes when you label fleshy mesocarp, it's not pointing as fleshy mesocarp, it's actually pointing at endocarp. So you make sure that it points there, then the juicy endocarp and the seeds. God help us in the exam. Then we go to specimen P, Q, and R. Specimen P is your thermometer, specimen Q, meter rule, specimen R is your quadrant. So I wrote down here, study specimen P, Q, and R and answer the following questions. The first one says, name one branch of biology which involves the use of specimen P, Q, and R. The answer is ecology. State one function each of specimen P, Q, R. P is the thermometer, is used to measure temperature or hotness or coldness of an object. Specimen Q is your meter rule, is used to measure the length of an object. Then specimen R is your quadrant, is used to determine the population of slow-moving organisms, or crawling insects. Name organisms each associated with the use of specimen R, that's the quadrant, and millipede, beetle, garden snails, striders, waterleaf, speargrass, and grasses. Anyone is correct. State 3 principles on which the use of specimen R for determining population density is based. The question can come anyway. So the principles include that number 1, the quadrant is used for the first thing for measuring slow running insects. 2, the quadrant must be tossed out up to, I wrote down here, at least 10 troops must be made. Then the last one, the last principle, the size of the quadrants depends on the organisms being sampled. I wish you the best in this forthcoming exam. I believe with God we have been able to look at some specimens and then analyze some useful questions. If you like my video, please share it. You can download, share to your loved ones, and then like my video, subscribe to my channel. And I know that you are doing that already. God bless you. See you in my next one. Bye.